All right, let's dive right in. We're going to be talking about bacterial traffic control, on a microscopic scale, of course. We'll be exploring these incredible molecular machines that bacteria use to basically manage their borders. Think about it. They have to import nutrients, export waste, and sometimes even deploy proteins as weapons. Understanding these systems, it's absolutely fundamental to grasping how bacteria not only survive, but thrive, cause disease, and crucially, how we might be able to stop them.
So here's the central question we're tackling today. How does a single cell manage what is essentially a fortified border? I mean, picture a gram-negative bacterium. It's like a tiny fortress, right? It has not one but two walls, an inner and an outer membrane. It has to get supplies in, get products out, all while it's under constant threat. So how on earth does it manage this incredible feat of logistics?
Now, you've all seen this 1,000 times in the lab, right? A petri dish full of colonies. But let's pause for a second. What are we really looking at? It's not just a smudge of growth, it's billions, literally billions of individual cells. And every single one of them is solving this complex border control problem every single second. The scale of this operation is just mind-blowing.
Okay, so here's our game plan. We'll kick things off with the border problem itself and what we're aiming to learn. Then we'll get into the energy cost of all this transport. After that, we'll look at how things get in, bacterial import, and then how things get out, protein export. We'll wrap it all up with a quick summary and think about what's next.
Right, first section, the bacterial border problem and our learning goals.
So, by the end of this, you should be able to really nail these points. We're going to discuss the role energy plays, tell the difference between transport across the outer and inner membranes, get a handle on the structure of the transport machinery itself, and you'll be able to give a solid example of a specific system. And finally, we'll explain the different secretory systems. Simple as that.
Okay, let's talk about power. Because, nothing moves for free in a cell. Moving molecules across a membrane, especially when you're fighting against a concentration gradient, say trying to pull in some scarce nutrient from the environment, that takes a serious amount of energy. So the big question is, where do bacteria get this energy?
Well, you can think of it like the cell has three different kinds of currency it can spend. First up, the universal standard, good old adenosine triphosphate, or ATP, which gets hydrolyzed by ATPases. Then you've got PEP, phosphoenolpyruvate, which powers a really clever system through substrate phosphorylation. And finally, you have the proton motive force, or PMF. This is just a fantastic bit of biology. It's an electrochemical gradients of protons across the inner membrane. The best analogy? It's like a microscopic hydroelectric dam, storing up potential energy that can then be used to drive all sorts of machinery.
All right, so we know how transport is powered. Now let's look at the pathways themselves, specifically for getting things in. For our gram-negative friends, this is always a two-step problem. First, you've got to breach the outer membrane, and then you've got to get across the inner one. Let's start from the outside and work our way in.
This diagram gives us a brilliant overview of that journey. See on the left? That's the outside world, the external medium. Then we have the outer membrane, followed by that gap, the periplasmic space. And finally, the last hurdle, the cytoplasmic membrane on the right. What we're going to focus on right now are the three main ways a molecule can make that very first leap across the outer membrane.
Okay, let's break this down using the table. Look at type A, the porin. This is your basic entry point. Think of OMP-C or OMP-F. They're just open channels, letting small molecules diffuse through. No energy cost, super simple. Now level up to type B, the specific channel. A great example is Lamb-B from maltose. It's still passive, no energy needed, but now it's selective. It's got a specific shape for a specific substrate. But then look at type C. This is the really cool one, high affinity active transport. This is for the important stuff, the scarce nutrients like iron or vitamin B12. A receptor on the outside, like FUA, grabs its target, but it can't get it in on its own. It needs help from this amazing protein called TON-B. TON-B is anchored in the inner membrane, and it reaches across the periplasm, couples to the FUA receptor, and then, powered by the proton motive force, it literally yanks the nutrient across the outer membrane. It's an absolutely incredible piece of molecular machinery.
So, the molecule is now in the periplasm. Great. But the journey's not over. Now it has to face the inner membrane. And that requires a whole different set of tools. Things like simple transporters, the ABC transporter family we all know, the list goes on. Now, for the rest of our time, we're actually going to pivot and focus on protein export. But it's really important to keep in mind that this whole second stage of import is happening as well.
Okay, let's completely flip the script now and look at the opposite problem. How do you get big, complex proteins out of the cell? This is absolutely critical for bacteria. We're talking about everything from secreting enzymes to break down food, all the way to full-blown pathogenesis, where bacteria are literally injecting toxins directly into our cells.
And to pull this off, bacteria have evolved this absolutely stunning toolkit of different secretion systems. We're not talking about simple holes in the wall here. These are complex, multi-protein machines. Each one has a designated number, and each is highly specialized for a particular job, a particular protein cargo, or a particular destination.
This diagram here is perfect for getting the lay of the land. Just look at it. You can immediately see there are two main strategies at play. First, you've got the two-step processes. See the type 2 and type 5 systems? They rely on first getting a protein into the periplasm, using helper pathways called Sec or TAT. Then they move it across the outer membrane. But then you have these other beasts, the type 1, 3, and 4 systems. These things are incredible. They build a continuous channel that spans both membranes, allowing them to shoot proteins directly from the cytoplasm to the outside in one single go.
So for any of those two-step systems, that first hop across the inner membrane is handled by one of two pathways, Sec or TAT. And the difference between them, the really fundamental difference, is all about the shape of the protein they're moving. The Sec system, which is the general pathway, takes proteins that are unfolded, like a long piece of spaghetti, and threads them through the membrane. They only fold into their proper shape once they're in the periplasm. The TAT system, on the other hand, does something completely different. It's specialized for moving proteins that are already fully folded up in the cytoplasm. It's a much bigger job.
Let's zoom in on that TAT system for a second. Its full name is the Twin Arginine Translocation System, and that name tells you a lot. It's built from three main protein components, TAT A, B, and C. And it recognizes its cargo by looking for a specific tag, a signal peptide that has two arginine amino acids right next to each other. That's the twin arginine, or RR motif. A classic example of this in action is in Pseudomonas aeruginosa. It uses the TAT system to secrete phospholipase C, which is a major virulence factor that absolutely must be folded correctly inside the cell before it gets sent out.
Okay, now let's put the spotlight on one of the most versatile systems of all, the type 4, or T4SS. This thing is just incredible. Why? Because it doesn't just transport proteins, it can transport DNA as well. This is the system used in bacterial conjugation. And get this, to build its channel through the cell wall, it brings its own demolition expert, a protein called VIRB1, which is a glycosylase that literally chews through the peptidoglycan layer. What's even more amazing is how modular it is. The whole machine is powered by a protein called VIRB11. And depending on what VIRB11 decides to interact with, VIRB4 or VIRD4, the whole machine can switch its function. It can go from building a pilus to actively translocating a substrate. That is molecular multitasking at its absolute best.
Whew. Okay, we have covered a lot of ground there. We've gone from the basic energetic costs all the way through to the nitty-gritty of some of these incredible import and export machines. So let's take a moment to pull it all together and nail down the key takeaways. All right, let's recap. What do you need to remember? First, protein secretion always starts at the inner membrane, and the cell has two choices, the Sec system for unfolded proteins or the TAT system for folded ones. From there, it can be a two-step process, finished off by the type 2 and type 5 systems, or the cell can go for the direct route, a one-step process using the type 1, 3, or 4 systems that goes straight from the cytoplasm to the outside. And the big picture here, every single one of these systems is a highly specialized, highly evolved piece of molecular machinery.
And that brings us to the final and maybe the most important question. We've just spent all this time looking at this incredibly intricate, essential machinery that bacteria absolutely depend on for survival, which means it's a vulnerability. So the question for you is, how can we jam the gears? Knowing what we now know about the structure and function of these systems, how could we go about designing new, highly specific antibiotics that target them? That's one of the biggest challenges and most exciting frontiers in microbiology today. Thanks for joining me.